Okay, so this is then the Year 7 Cells Knowledge Test. Right, now what you do now is you watch this video, you fill in the form, fill in the test paper that you've been given by your teacher, right, and then what you then do is then you, at the end of it, you've got to learn it, and then you've got to retest yourself. So it's all the facts and all the information that you then need to know to pass the test. Right, so what we've got here is name all the parts of the animal and plant cells and what are their functions. So number 1, right, which is up here, it's not particularly very clear, is a chloroplast. I'm not going to write these in very well. Chloroplast. Okay, and the job of that is photosynthesis. Okay, which is the production of energy from the sun. Number 2 is it's this one here, a ribosome. Right, and the job of a ribosome is protein synthesis. Right, don't write makes proteins, you write down protein synthesis, which is basically the same thing, but it's the proper way of saying it. Number 3, what we've then got is we've got a mitochondria. Right, and the site, mitochondria is the site of aerobic respiration. Okay, which is then where glucose is then used to produce energy. Number 4, let's do a bit of green again. Right, number 4 is the cytoplasm. And the cytoplasm is the site of chemical reactions. Okay, it's kind of like the jelly-like substance that's in there. Bit of orange. Number 5 is the nucleus. Okay, which is this one here. Right, the nucleus, it contains DNA. What it kind of does, it kind of controls the cell. So it provides all the information to make sure that the cell is working correctly. Number 6, we've got a cell membrane. Right, and what that does, it controls what goes in and out of a cell. Number 7 is a cell wall, which keeps the structure, and it's made of cellulose. And number 8, go back to green, is a vacuole, which then contains cell sap, which keeps the cell or the plant upright. Describe the method for setting up a microscope. Right, so it's kind of like 4 stages to it. Right, firstly, you do the nose piece to the lowest magnification. Right, that's the shortest one. All right, so that is then the nose piece lowest, and it's the objective lens that you're turning around. Number 2, right, put the slide on a stage. Number 3, focus using the large dial. And number 4, draw it. Okay, it's relatively straightforward, right? So really you just get your microscope, right, turn it to the lowest objective. Best thing to do is put it down right as far as it will go, as far as possible. So then what you're doing when you're actually focusing, you're moving away from it. Put the slide on, focus it, draw it. Describe the method for preparing an onion slide. Right, now this is actually one of the GCSE required practicals, so it's dead important that you know it. So number 1 is peel off the epidermal layer, dermal that is, layer. Right, what that means is there's like a really thin kind of piece of skin in between each one of the layers, and when you peel it off it looks like skin, right, and it's dead thin. Number 2 is put it on a slide. Okay, and possibly just add a tiny little bit of water just to moisten it a little bit. Number 3, add iodine, and what that does is it stains it. Number 4, put on a cover slip and press. Now I don't mean press really hard, because the cover slip is like a very thin piece of glass. But the idea of that then is just to flatten it all out, get rid of excess iodine and get rid of any air bubbles. Number 4, give 2 advantages and 2 disadvantages of light microscope and electron microscope. Right, so an advantage of a light one is the fact that it's cheap. Right, it's also light to carry. A disadvantage is it has a low magnification, right, you can get up to about times 400, something like that, and it's got also a low resolution, right, which means you can't kind of distinguish between 2 points particularly very clearly. Then you go to the electron microscope, and what you do is you go that way, so you go, it's got high magnification, it's got high resolution, right, which is kind of the opposite to the disadvantages. The disadvantage is it's expensive, okay, and it's also very big and very heavy, okay, which is obviously not good. You know? How would you calculate the magnification if you know the eyepiece lens? No, that's probably a spelling mistake there in the blue. Okay. How would you calculate the magnification if you know the eyepiece lens magnification and the objective lens magnification? So what you do is you do eyepiece magnification times by the objective magnification equals the total. So if the eyepiece is times 10 and the objective lens is times 40, 10 times 40, the total magnification would be 400 times. So name 3 animal and 3 plant cells and explain how they're specialised. Right, so I'll write down muscle, sperm and nerve. Okay, so we'll just do these first, right, the animal ones. So a muscle, the main thing it can do is it can contract, and it also has a lot of mitochondria for giving it energy. Sperm, it's got a tail, it's got an acrosome layer right in the head, which is part of the way that it manages to break into the egg, right, and it also has a lot of mitochondria in the neck. Okay, so where you've got the sperm, what you've got is kind of like a neck bit there and the tail. Here you've got a lot of mitochondria for the energy, and here is then the acrosome layer which helps break in. And another important one that you've got, okay, let's do it in a different colour, is here you've got the nucleus of the sperm and it's got half of the chromosomes. So half from the sperm, half from the egg. Nerve is long and it can conduct electricity. Right, plants. We've got root, hair, we've got xylem, and we've got phloem. Okay, right now, root hair first. The whole point in a root hair is the fact that it is a large surface area. Right, so what you've got is you've got the normal plant cell, and the root hair has got a big long thing that sticks out the side to increase the surface area. Xylem, xylem, you've got to be able to spell this one. It's one of those words in science that you must have the ability and must be able to actually spell it correctly. Okay, and what it does is it carries water, minerals up a plant. It's just one direction, right? And the fact is that the xylem is dead. They're just like tubes of lignin, right, which is then dead. Phloem, it carries glucose plus other things, 2 directions. Okay, so it can go anywhere around the plant, right? It's living, and it also have things called sieve plates, right? And what those sieve plates are, they're on the end of each individual cell. Right, definition of diffusion is the movement of particles from a high concentration cells, that's concentration, to a low concentration, down a concentration gradient, and is passive, which means no energy. Okay, so it's the movement. So if you think somebody walks in a room with perfume on, right, where the perfume is a high concentration. So what it does, it moves across the room from a high concentration to a low concentration, which is called going down a concentration gradient, and passive means no energy is needed for it. Question number 8, right? What is aerobic respiration? Aerobic respiration is where an organism releases energy. Okay, right. The equation for it is glucose plus oxygen gives water and carbon dioxide. Okay, so that's the word equation for it. Question number 9, name 4 unicellular organisms, describe their appearance. Right, so we'll go for a bit of orange on this one. We've got amoeba, we've got bacteria, we've got euglena, sorry I'm spelling that wrong, euglena, and we've got fungi. So an amoeba is single-celled, and sometimes it says that it kind of looks a bit like a false foot. Bacteria is single-celled again, right, and some of them have flagella. Euglena are again single-celled, but they have chloroplasts. And fungi, they are bigger than the others. They are still single-celled, right, and they are spread by spores. Right, key definitions. The smallest functional unit of an organism, the building blocks of life, cells. A measure of the number of particles of a substance in a given volume is the concentration. So if you put more kind of Ribena into your cup, you increase the concentration because there's more particles. How much bigger an image appears compared to the original object is the magnification. An optical instrument used is a microscope. Living things are all organisms. The ability to distinguish between 2 points in a specimen is a resolution, so an electron microscope has a much bigger resolution. A cell whose shape and structure enable it to perform a particular function is specialised cells. We went through 6 before. Consists of one cell, is unicellular, or it can be single cellular. Right, now that is the Year 7 cells topic. And again, that is the minimum that you need to know. So what you do now is you go away, you make sure this is filled in, you go to the blank version of this, right? And when you find the blank version of this, you watch the video, pause at each individual time the questions on, write down your answers, right? And if your answers are correct, fantastic. If they're not, play the video again, right? And then keep repeating it all the way through.